Okay, so yesterday we finished with some kind of the table of gene expression, which we prepared and filtered outliers and normalized the data set and so on. And today we'll talk about what you can do next. And yeah, so let's open this data set right now, like the one that we finished with. So for this, yeah, like as yesterday, the slides are available in Google Drive. Also in Google Drive in day 2, there is a file called GSC something filtered. So please download it and load it into Phantasus. So this is a dataset that we finished with yesterday. So please go to day 2. There will be this GSC filtered GCT file. Download it. Yep.
Sorry, could you hear me?
Sorry?
Hi, could you hear me?
Yeah, but not that loud.
Okay.
Oh, yeah, now it's better.
I have one question. Maybe I should. Oh, great. Well, okay. Could you please explain or elaborate on which files could we process with Phantasus? I mean, it's kind of used only for geo datasets, or we could use it for our self-obtained data somehow.
Yeah, like we'll talk. So now we start with public datasets, and later, like in the second part of today's lecture, like today class, we'll talk about how to load your data into Phantasus. So yeah, we'll go back to it a little bit later.
Okay,
but for now,
thank you.
Yeah. Yeah, and you should have the slides in day 3 folder. Nope, actually no, sorry. Yeah, now you should have the slides for today as well. And so please, like everyone, load the dataset, and well, raise your hand if you have this kind of table. Yeah, and can people say on Zoom say okay, we have this, or that you don't
yes,
have it?
We have it.
Yeah, great. Okay, so this is the table from yesterday. So it's oh, that's like very big raising the hand. Okay, so this is a table from yesterday. So just to remind you that we actually there are some slides on this. Nope. No. Yeah, usually I'm doing it in the same day, like continuing. So yeah, to remind you, yesterday we filtered outliers, which were like the first samples. We also log normalized the data set, and we filtered out lowly expressed genes. So now we have these 12,000 genes. Like everything is nice, and actually we can try to do some analysis. And the first type of analysis is very simple, and like the most used type of analysis in bioinformatics in gene expression data analysis. And this is so-called differential expression, where we try to find genes that are differentially expressed, have different expression between two conditions of interest. So for this, in Phantasus, we have this tool called Lemma, and Lemma, maybe some of you know, who a bit familiar with gene expression analysis. So Lemma is an R package for like linear models. So L and I probably stands for linear, and I guess M is for models, and for MA is for microarrays. This is a package for, for work, which was developed originally to work with microarrays. So this is like one of this package, like originally from 2000 and something, like very early, like basically almost the start of the bioconductor library. And yeah, like one of the features of Phantasus, like why we do like it and a lot, is that this type of analysis you can't like re-implement. No, you can re-implement, but it's very hard to re-implement it like any other language. So basically, that's how we use, like how we interact with actual R packages without redoing all the code. So yeah, let's do this. So let's go to Phantasus. Let's do tools, differential expression, and lemma. Well, for differential expression, you would like to have two sets of well, samples to be compared. Well, usually, at least. And for this, we'll select samples based on treatment. And so we'll be comparing untreated samples as class A, like it's a baseline, so as a control. And we'll compare it with LPS samples, which is LPS treated samples, to see what genes are differentially regulated on macrophage activation. So when you do this, a table should appear, like with all these numbers. Oh. Yeah, and actually let's do click on this T column to sort by it, and there we'll see. And you'll see that, well, all the genes on the top are very red in this LPS condition and blue in untreated samples. So which is like what we wanted, at least what I wanted for you to have. So basically, these are genes that are upregulated, that have higher expression in LPS samples compared to untreated samples. And like in the bottom of this list, you have genes that vice versa are downregulated, that have lower expression in LPS samples compared to untreated samples. And yeah, and in this table, there are multiple values. So let's go over them one by one, or most of them. So let's start with average expression, well, AFEXP, which non-surprisingly is very similar to mean column that we had before, so the average expression. So in this case, like in particular in Phantasus implementation, like how it interacts with lemma, so this average expression is average expression in this in the selected samples. So not all 12 of them, but only these 6. And yeah, you can check that indeed, like for these top genes, like the top gene, it has expression on average about 10. The second thing, like the important thing, is log FC, and log FC is log fold change, and actually log 2-fold change. So it represents well, fold change, like gene expression fold change in logarithmic scale. So what that means that this is log 2 scale, and actually it's important that for lemma, the normalization that we did before, log scale normalization, is actually important. So lemma, as I was trying to tell you yesterday, like linear models really like to work with data that has normal distribution, so that normal noise and so on. So and this works only for gene expression data in log scale, not linear scale. So yeah, basically we did the log scale, and now log fold change is actually lemma doesn't change anything. So this is just in our case, because the data is already log scaled, it's just the average difference in these numbers in LPS samples compared to untreated samples. And actually, let's look at the actual numbers. So like in LPS condition, we see that there
Excuse me,
yeah.
can I ask,
Yeah.
what is a T?
Well, it's almost, it close, like it will be on our list next. So, yeah, so first of all, like log-fold change. So if you see that like LPS treated samples have average expression around 14, untreated samples is expression around like 6, and like 14 minus 6 is 8, which is the same as log-fold change that we have here. So it's nothing magic about it. So just in our case, it's average difference between the samples. And of course, like if it's positive, then the gene is upregulated, and if it's negative, it's downregulated. So now let's go to p-value first. So again, not again. Who knows what is p-value? I hope that everyone knows. Not. Yeah, like can anyone tell me what is p-value? Okay, well, sounds pretty all right, but very long. So yeah, so basically for this we have some kind of like in the p-value, important part is a statistic value. So this is like some kind of single value describing like some situation, and we have this null hypothesis, which kind of like our hypothesis that nothing happens, like which can be different in different cases. And p-value is the probability that if you have this null hypothesis correct, to have on like the value, this our statistic value, at least the same or more extreme than we have. And so there are two parts in this. Important parts, like one is statistic and the other is null hypothesis. So what would you guess would be a null hypothesis for differential gene expression test? Yeah. So indeed, the like very like straightforward and natural null hypothesis for gene expression data, like when we compare two conditions, is that that we say that average expression is equal in both, like in our case, untreated samples and LPS treated samples. And otherwise, you can say that like null hypothesis is that log-fold change is 0, like true log-fold change is 0, not the measured one, but the true one. And also, well, null hypothesis also like includes something about the model, like how the data is distributed and so on. So, and this can be different in different models for gene expression data. And in our case, because we have this linear transformation and some other like magic by lemma, what it does is uses like something very similar to t-test. So basically, we assume that we know the gene expression is normally distributed in one condition, and normally distributed in the other condition, and well, null hypothesis that both this normal distribution has the same like variance and the same average value. Yeah, and there is some magic involved that the variance is not estimated. So usually in standard t-test, you estimate like variance based on the like one particular well, again, sample, but not like statistical sample. And in this case, like for gene expression data, because there are no too much replicates, many methods try to infer this variation, like variance, in more complicated ways. But basically, yeah, it somehow estimates the variance and then tries to compare them like the log-fold changes. And this is it's very similar to standard t-test. Basically, it's called actually moderated t-test, and the statistic that is calculated is like moderated t-statistic. Well, moderate particular like some basically moderated kind of magic, how it's moderated. But in the end, there is t-statistic, which is distributed like as a student, like with a student distribution. Oh, yeah. So yeah, the t here is t-statistic, like moderated t-statistic. And what you can tell is that, and the p-value is a two-tailed test. So basically, it's the p-value is very low, both if you have highly positive t-statistic and highly negative t-statistic. And the sign of t-statistic is the same as the sign of log-fold change. So when we started, by decreasing t, as we have here, we have all the genes that has positive value of t-statistic, means that they have positive log-fold change, and all the in the beginning they have like very significant p-values, and in the bottom they have also very like highly negative t-statistic values, and they also have very low p-values. And in the middle part, the t-statistic is not that big, like by absolute value, and the p-values are very close to 1. Okay, and who knows what is AdjPval column?
Adjusted for multiple.
Yes, it's adjusted p-value, but what adjusted for what? And
Multiple hypothesis testing.
yeah, the size of our genes seems almost right, but there is a special word for this. Yeah, multiple comparison. So yeah, this the p-value column is so-called like nominal p-value, basically p-value that comes from t-test. But as you, I hope, all know, like from the previous semester, when you do like multiple comparison, in particular, here we have 12,000 comparisons. So we definitely want to correct this for like multiple comparisons testing. So yeah, and adjusted p-value here is, and who knows like what methods for well, multiple hypothesis correction? Bonferroni, good. And is there anything else, like any other methods? Yeah, there are many of them.
Benjamin Hochberg.
Yeah, Benjamin Hochberg is also the good answer. Well, there are other comparisons as well, but this particular, like in bioinformatics, people don't like, especially practical bioinformatics, people don't really like Bonferroni because it's very strict, and particularly if you have like 10,000 genes, like it will be effectively multiplying all the p-values by 10,000, which is pretty strict. And what people usually use instead is Benjamin Hochberg correction, which instead of trying to make to maintain family-wise error rate, means that probability that like everything in the top, like all the top is correct in the same time, it tries to maintain false discovery rate, meaning that what proportion of the prefix can be false positives. Yeah, but yeah. So in our case, this is Benjamin Hochberg corrected p-values, and well, you can use the threshold like for this. If you want to select some kind of statistically significant genes, you would want to select by adjusted p-values here. Oops. Okay, any more questions about this part? Okay. Okay. Yeah, and I would also like recommend you. Do I recommend it? Yeah, you can save at least the data now. But after that, basically what you so this is one of the steps that you like again to the differential expression test is one of the more straightforward approaches. And usually, if you analyze your like some kind of data set and you want to speak with your like collaborators, to like people who did the experiment, if you are not them, so usually send them the table of with differential expression results, so they can also see them. And well, GCT file and we'll try to open it later in like Excel or whatever. So it's like simple text format that people can open. However, also like for sharing the results, there is another feature that very useful in Phantasus is sharing the results by link. So let's do the following. So let's go back to Phantasus. In the file menu, select get dataset link.
I have a question.
Yeah.
Okay. Yeah, I have a question. Could you please come back on one slide, on previous slide?
This one?
I guess this picture is okay. Yeah, or yeah, I just have a question about, I guess, 10th or 15th gene, because he has not that colorful red color for LPS, and still it has a great difference in the numbers. How come we have a big difference in numbers, but still we have no that much difference in colors in heat map?
Which again, Jin?
I guess I can say the name. For example, PYD04, I guess.
PY.
Well, I'm not really sure. I guess I could check it on my PYDC4. Yeah.
Okay. So,
for example.
Yeah. Well, it looks like a pretty nice differential expression.
Oh, I'm sorry. The next one. The next one.
BST1.
So, yeah. So he has difference, but I would say that this difference is not that much as, say, the next one gene has. And why then we have this gene higher than the next gene?
Well, actually, I would say like otherwise. So there are two, and actually, let's do. Let's see. Hopefully, it will work. So there is important difference, like one in statistic, that you have p-values like that somehow represent the statistical significance, and also like effect size, which represent I know, like some kind of physical difference. And usually they somehow correlated. So basically, if you have on average big log-fold change, well, you'll have highly significant p-value. However, it's not that linear, because because aside from the fold change as itself, in the t-test and like any other test, variance is important. So, for example, you can have small log-fold change difference, but the expression is very tightly has very small variance in one sample, like in one condition, and very small in other. So, like compared to this log-fold change, this average difference compared to the variance in each condition will be very big. And on the other hand, you can have seemingly high log change, so high difference of expression, but the variance, like noise, is also very high in this like gene. So the basically, and in this case, it won't be significant. So basically, the log change can be like similar to the noise itself, and you can't really say if it's different or not different. So and in this picture, so one of the actually, do not adjust it, but nominal p-value. Yeah, it's not very different, but still. So basically, we have usually we have like if you have high log change, we have very low p-values. So this is like log scale. However, there is still like with you can have log change of I don't know 7.5, and you can have p-value 10 to the minus 10 and also 10 to the minus 4. And this means that the variance of this gene is smaller than variant like than here, or like the noise here is higher than the noise here. So that's why sometimes you can use you can see that log change is like higher in one case, but the p-value is not that significant. So yeah, this can happen. However, like overall, the result will be more or less correlated. And usually, and like as I said, so there is like this effect size, in our case, log change. There is statistical significance, but I would like to remind you, like in particular, this course is about so that you as a computational biologist, not bioinformatician, has to try to find genes that are biologically significant, not statistically significant. So for this, well, okay, you can have like many samples and can see like p-value less than 0.05, but the log change won't be high. Well, it doesn't mean that probably you can remove this gene and nothing will happen. So in that sense, statistical significance can be more important, like more better for ranking than like log change. However, also usually, and actually like in this picture, like in this volcano plot, you can see that there are two thresholds. One is for log change, one is for p-value, but the other is for log change. So basically, in particular, if you have well, significant p-value, but low log change, well, probably it's kind of statistically significant, but not that biologically significant. So, and again, it can have high log change, but not statistically significant. That probably is also not significant. So while, and from bioinformatics perspective at least, well, so in this part, like blue part, like in actually like a blue arrow, like an avatar, these are like not significant, like either statistically or biologically. However, like remember that it doesn't mean that the red genes are biologically significant. So still, like if you do knockouts of these genes, even they have like even this part, they can like nothing can happen. Like well, something could happen, but like not in your phenotype. So so there are a lot of genes, and probably they just happen to change for whatever reason.
Okay. Thank you very much. Still, didn't we sort for T and not for p-value? Or in this case, it's the same?
Well, in our case, like for lemma, ranking by T and ranking by p-value is different. Just so I prefer ranking by T because all the genes that are significantly upregulated are in one end and significantly downregulated in another end. And you'll actually see, well, let's do the bird's eye view. So, and we'll see later today why it's in general useful. So basically, we can see here that these genes are highly, like significantly upregulated. These genes are significantly downregulated. And if we sort by, if we look only on the genes that have positive log-fold change and rank them by p-value, it will be the same ranking as by t. So in this case,
Okay.
While log-fold change is ranking is different from ranking by p-value, ranking by t among the genes that has positive log-fold change is the same as ranking by p-value.
Okay, thank you.
Yeah. Okay. Yeah, and I was telling about sharing the results. So another useful feature is, so you can get this dataset link, copy it, and send it to your collaborator, and hopefully it will work, and you'll get the same kind of results. So usually it's pretty convenient to discuss it, to do some analysis and discuss the results. Okay. Well, so we did the differential expression analysis, like was one of the foundation of gene expression analysis, and it was pretty simple. However, and now the question is, well, what to do next with this many genes that are differentially regulated, like statistically differentially regulated. And one of the approaches for this, also like more or less standard, and is pathway analysis, or pathway enrichment analysis. And the general idea is that, like, again, if you look at the number of genes that are differentially regulated, it will be like pretty high. There will be a lot of genes with some weird names and with some weird functions. Maybe, well, you can go over like the top of this, like this CD38. Probably you can open Wikipedia and read about it, but then there are some. Well, you can see like IL-1B, like some cytokines, that you can probably know, but overall you can't really go over all of these genes one by one and try to comprehend the situation in general. And another thing is that genes, like so we have a lot of genes, and also like even a single gene can have multiple functions in different contexts and so on. So it's useful to go from from genes to pathways, or like to some kind of biological processes. And in this case, like pathways will be the genes that are involved in the same biological process. And then like it's much easier to reason about processes, biological processes, than about particular genes. Sorry. So, well, let's try to do some kind of pathway enrichment analysis. And for this, we'll use this MSigDB web tool. So let's go to this website. Oops. You can go by the link or just Google it. Okay. And it will ask you to log in. So you can put my email there for now, but you can register. It's free, so should be no problem. And there should be interface like this. Okay, who have this interface? Great. So now let's do the following. So let's go back to Phantasus. And the questions that we'll be trying to ask is, okay, we have this like a lot of upregulated genes. Well, let's select like some kind of like top 200 genes, or like 250 genes, and we'll try to understand what biological processes these genes are about. So let's select like roughly 250 genes. Oops. Well, you can do it like I do, just by scrolling a few screens, or you can do it like properly and use the filter menu. but it doesn't really matter much which way you would do you do it. But in the end, so we have this top upregulated genes selected. So let's copy gene symbols. For this, like click copy, like right click on the gene symbol column and select copy selected values from gene symbol. And paste these genes into MSecDB2. Okay, is it working? Yeah, it's important to right click on the particular column, not just anywhere. Yeah. And for this MSecDB2, let's select the following. So, well, first of all, let's select species. So in our case, this is, okay, I have this on the slides. Probably in the next iteration, I should ask people what organism is this dataset. But yeah, this is mouse dataset. So this is mouse genes. And actually, one useful thing to understand, like about gene names, like gene symbols, that will be usually useful, is that the like mouse gene symbols has the form of well, they start from the uppercase letter, one uppercase letter, and then like lowercase letters. And human gene symbols are all uppercase. And well, so usually it doesn't matter much, but sometimes it's useful to understand which genes are which. Okay, so we selected mouse, and next we'll select the so-called like gene sets, which will be comparing our top our gene set with. And these gene sets describe different processes, different biological processes. And well, let's for now select some hallmark gene sets and canonical pathways, for example. And click compute overlaps. Yep, and you should have this kind of table. Yes? Yes. So this is one of the well, like general approaches for gene expression for pathway analysis or like gene set analysis. And in this result table, we have, well, each row corresponds to one particular pathway or one particular gene set. So in our case, like this first result is genes that are involved in interferon gamma response. Then the genes that are involved in interferon alpha response, and so on. And next, well, in here we see that this gene set, this pathway has 200 genes, 97, and so on. Next, we see p-value, which is, actually, let's talk just about overlap. So overlap is the number of genes, well, that overlap between our query, our like top upregulated genes, and this each particular gene set. So between our like 280 the genes that I copied, 70 genes are contained, like intersect with this interferon gamma response pathway. And what you probably could guess is that this is like a big overlap, and probably it's not random, and it's indeed does have very low p-value. Yeah, and actually, if you scroll a bit down, you also can see that which particular overlaps you have. So in this table, we have our pathways, our gene sets, like reference gene sets, and our genes. And we can see like which genes from our query are contained in this pathway. So we see that in gamma response pathway contains these genes like IRF7, STAT2, and so on. So if you somehow familiar with immunology, so these genes will make sense to you, like at least the appearance in this pathway. Okay. Yeah, and in this type of analysis, it's important, well, actually in any kind of analysis, it's important to understand how the p-value is calculated. And in our case, the Fisher exact test is used. So who is familiar with Fisher exact test? Not like hypergeometric test, maybe. No? Okay, only one person in the room. And on Zoom, anyone? Okay, but basically the idea is the following. So we have our query, like our query set that we are trying to find which gene sets it does overlap. We have this some particular gene set, well, in this case, like reactome immune system, that has overlap of 50. And the null hypothesis is that the overlap is obtained as random, like in random. And for this, there is some important parameter called the universe. So basically what, in general, what is the universe of all the genes that you are selecting from. And basically, the idea is that, well, in our case, the MSecDB considers a universe of like almost 50K genes. So in this universe, if we have, so which pathway? This example is for this interferon alpha beta signaling pathway. So it has overlap. Well, with this like 50 genes probably is not random. But like let's try to understand like with 70. So we have this universe of 45,000 genes. We have this gene set of 64 genes, and we have like our query gene of like 196 genes. So if these genes, like if these two gene sets are independent, then you can calculate the like average number of like expected number of overlapped genes, and it will be around like 0.27, even like less than 1. So and indeed, and we see like 17, which is much bigger than this 0.27. So I want to go into details how the exactly p-values like for distribution appear there, but basically, yeah, it's can we obtain this? Like what is the probability to obtain this kind of overlap if both gene sets are independently drawn from this universe?
Sorry.
And important part here is that the universe, so that MSecDB uses is 45K genes, and the universe that we had in our analysis is actually 12K genes. So we selected only the top expressed genes. So it's so we didn't use like these 30,000 genes. So and in particular, there can be like additional overlapping genes and so on. So in our calculations, instead of using like in proper calculations, we should have used not these 45K genes, but these 12K genes, or actually intersection. Like even better, it would be like an intersection between like 12K genes and these 45K genes, which will be even smaller. But now, if we consider that we have universe of 12K genes, the average overlap will be not 0.27, but 1, which
Excuse
is,
me.
well, yeah.
Could I ask? I don't understand where we take 64 number and 17 actually.
Yeah, yeah. Well, this is the last row of the results on the slide. So the 64 is and can you see how I put my hand on the screen? Can you actually see my camera? Hello?
Yeah, we can.
Yeah. So 64 here is the number of genes in this reference gene set. These are the genes that some people said that they are involved in interferon alpha and beta signaling. So this is some kind of curated gene set that is present in the reference dataset. The 17 is the number of genes that, and we have this approximately 200 genes in the query. So we upload it into MSigDB, like 200 genes. And between these 200 genes and these 64 genes, there is an intersection of 17 genes. And the statistical, Fisher test says that obtaining such big overlap, like overlap of 17 genes, is very it's hard to obtain in random, assuming if you have this universe, total universe of 45,000 genes, and that both these 64 genes and these 200 genes are obtained and randomly from this universe.
Thank you.
Okay. Yeah. So coming back to this point, so the point here is that when we decrease our universe, the random overlap will be higher, expected overlap will be higher, and while the 17 will be still significant, it actually will be less significant, less statistically significant if you do all the things correctly. So in this type of analysis, when you do it, be aware of these kind of problems. And usually, in this case, when you see p-value of 10 to the minus 20 or 10 to the minus 10 even, so it doesn't matter. It still will be significant. However, in MSigDB tool in particular, if you see adjusted p-value of 0.01, which is less than the standard 105, it likely won't be significant if you do everything correctly. So be aware of this. So sometimes you have really random genes, not random, but there are not any significant overlap, and there will be some slightly significant results in MSigDB, but it's actually false positives. So, yeah, and talking about the pathways and what are they and where are they coming from. So one of the pathways in the example, in the slides at least, and probably we have it here. Yeah, reactome alpha beta signaling. Okay. Or it actually does work. So at some point it is broken. So basically, in the MSigDB tool, you can click on the gene set, and you'll have some kind of description. And in particular, for reactome datasets, for example, you'll have this link to reactome database that you can click on, and there will be some nice, relatively nice pathway, pathway picture that you can look into further. Mhm. Yeah, and you can look into different interactions here and see why are they here, what are the like PubMed references, and so on. So, well, you can explore it a bit further to understand what are these pathways about. Yeah, and another thing that you can do. So at this point, we like from the MSigDB tool, we understood that okay, this intron alpha beta signaling pathway is something happens with this pathway in our dataset. So it has at least like 17 genes significantly upregulated in our dataset, like 17 from 64. And the question that could appear, you could ask, is like what happens with all like other like 40 genes? So are they also like upregulated, either downregulated, or are they like not regulated at all? So to get this overall picture, so let's find what are the genes in this gene set, and we'll see. We'll look at them in Phantasus. Okay, so we close this rectangle. Click on the text, like in the download gene set, click on text. And you can use like any other pathways that you like from the top results. Okay. Open it in like in any text editor or whatever, and copy these genes. And paste it into Phantasus. Yeah, one thing to notice again is that the genes in this pathway are in uppercase, right? This means that they are human genes. Like only human genes are named in uppercase. Like all other animals are like first letters uppercase and then lowercase. So these are the human genes, and what it means actually is that the genes in the MSigDB, at least like in particular in Hallmark, we are created for human. So this is also like one thing to keep in mind when you apply this to other organisms. So usually, like for mice, it's kind of similar, but if you have some, like for example, some weird animals or even like plants or like bacteria, so you should find some other database that was curated for keeping this organism in mind. Okay, but yeah, we copied these genes, and yeah, let's skip this, and let's go to the fit to window again. So something like this should appear. Who has something similar?
I think I have.
Good. And are there like any problems? Have you been able to download the gene set, copy the genes, and so on? Okay. Well, hopefully it works for most of you. So what we can see from this kind of picture, that actually not that we only have a bunch of genes that are significantly upregulated. We also have this number of genes that are not maybe that significant, but still upregulated. And well, some of the genes are downregulated. But overall, we can see this trend that like you can see that this pathway is mostly upregulated. And one of the ways that people use to visualize it and put into papers is in an enrichment plot. Sorry. Yeah. So let's, oh, like GSA plot. So let's go to tools, plots, GSA plot. And well, let's run by T. Yep. And something like this should appear. And actually, let's just switch to vertical. So maybe some of you have seen this type of graphs in publications. So they usually used when you want to well, visualize some particular pathway that it's a regulation in one particular data, like in particular different expression comparison. And so this works as follows. So these bars, like ticks here, are correspond to genes. So so you can see that actually these ticks are very similar to these orange lines. So it's actually not coincidence. So basically, we have the same ranking by T, and on the genes from the pathway, like from the gene set, are selected here. So now we have this curve that basically goes, well, in this case, in this format, goes to the right. So now we are going over the genes one by one. And if you have our gene, so we should, let's get it 100%. Yeah, so we look at like gene CD38 because it's not in a pass, it's not in our gene set. So we go to the left. Well, you can't really see there, but we go to the left there. So the next one is also, so we go a bit to the left and so on. Next, we see the genes from the gene set, which is marked here. And on these genes, we go right, to the right, and proportionally to the T value. So like for, no, I don't see the numbers there, but proportional to T value. Then we go a bit towards the left, again, like for 3 genes. Then we have another gene from the gene set, so we go right and so on. So, yeah, probably I should have explained differently. Yeah, let's go to this slide first. So this is the same picture, but well, horizontal. So we are going to genes one by one, and if our gene is in gene set, we go up, and if it's not, we go down. And now what happens if our gene set has a trend, like has more genes in the left part than the right? So the curve will be going upward and then downward. If you have the genes that are mostly on the right but not on the left, we'll go first down a bit and then we go up and we come to 0 each time. And if you have some random gene set that has, well, genes appearing all over this ranking, so we'll have this curve going up and down, and it will be going up and down. So so this is the curve, and this curve is used for this so-called gene set enrichment analysis that allows you to calculate some kind of p-values. And again, for these p-values, there is a null hypothesis and the statistic. So in our case, null hypothesis in this particular analysis is that the genes are distributed uniformly across the ranking. So and the score, the statistic that we'll be using will be this GSA score. And well, we have this GSA curve, and the score is defined as most extreme, based on the most extreme point in this curve. So in this case, when it's like high trend toward upwards, so we'll get the top point, and the enrichment score at this point will be the enrichment score of the overall pathway. And in this case on the right, we'll have the like most extreme point will be at the bottom. So this -0.7 is the enrichment score for this pathway. And the thing is that, well, if we have positive, like genes trending toward the left part, we'll have positive enrichment score. If we have like highly positive enrichment score, if we go genes has trending towards the right end, will have negative enrichment score, like highly negative enrichment score, or relatively negative enrichment score. And if you have some random gene set, we'll have, well, some kind of enrichment score, not too high, not too low. Okay, and well, using this definition, we now can calculate p-value by comparing this enrichment score with random gene sets. And because our null hypothesis was that the gene sets are distributed uniformly, so we can have this so-called empirical p-value, basically not analytical p-value, based on random sampling of gene sets. So basically, again, sample some number of gene sets and compare like how frequent is this or more extreme enrichment score in our random sample. And well, we do this we can do this like in our case. Yeah, we did this for one particular pathway, right? So we put this enrichment plot and actually calculate the p-value, but usually, well, not usually, well, yes, usually it's done again for some kind of pathway collection. It can be like MCDB, for example, pathway collection, or like gene ontology or reactome, like any other. Well, and again, later you have to normalize for multiple hypothesis correction. Yeah, and one thing about this kind of analysis is that, well, it was developed like in 2005, yeah, and used a lot. However, this calculation of these random gene sets is not that fast. Well, actually, not fast at all. So, but luckily, thanks to these two guys, yeah, not Elizabeth, sadly. Thanks to these guys, we developed a tool like for fast gene set dilution analysis that we can do. We can like use in particular in Phantasus. So let's go back to Phantasus and do a pathway analysis and perform FGSA. And let's select a smaller database, hallmark database, this for mouse clues for mouse. And interest here means that these genes are, well, this database is developed for interest gene IDs. So yeah, run by T, and we have interest IDs in the gene ID column. Okay, and if it says something like this, please do use the collapse tool. Okay. Sorry, just need to check something. Pathway analysis, perform FGSA, MCDB, by T and gene ID. Yeah. So if you have this error, this is like some minor bug in Phantasus. So please use the collapse tool, like as we did before. So and collapse them by gene ID. So like, yeah, and you have this kind of table. Again, it's the same as, well, almost the same as we saw before. So this is our pathways, our p-values, and you can click on the pathway and see all the genes here. And what you can do next is like copy these genes and paste it here. And you can see that indeed these genes are, well, up. Indeed, the gene set is upregulated in our data set. Okay. Yeah, an exercise is to do an enrichment plot, but we won't be doing it. Yeah, let's do a couple of more things and go for a short break. So yeah, please copy some kind of genes from gene set, or like from either like reactome pathways that we had before, or like from the RGC results. Doesn't matter much. So we see that indeed these genes are upregulated in this kind of comparison, in this particular comparison. But another question that people that you can ask is what happens with these genes over all 4 conditions. And like, so for this, there is useful tool to visualize this is using the like an average profile of the genes. So for this, you have to select all the genes from the pathway, select all the samples, and add like mean profile, and adjust. So usually it's useful also like to adjust to use so-called Z-score transformation for the gene expression, so that they have 0 average value. So we do this score transformation, and we'll see something like this. So we see that this pathway, I forgot what the pathway was. IL-6 JAK STAT3 signaling is actually, and let's add some labels. Yeah, so while the overall gene, individual genes can have different patterns, but the overall, it looks like it gets a bit upregulated on interferon gamma, even more upregulated on LPS, and even bigger upregulated when the treatment is like both interferon gamma and LPS. So this is also like a useful type of visualization that sometimes, nowadays, when you have a lot of conditions, can be very useful to have. Yeah, and the last part in this, last thing in this part is let's use this Enricher website. And the Enricher is a website, well, in some sense similar to MSecDB. It's for enrichment analysis. Well, you can guess from the name. And it does operate in similar manner. So for this, we want to select some genes, like 200 genes, either upregulated, maybe downregulated, and use the tools pathway analysis and submit to enricher. And select the column with gene symbols to be gene symbol and click submit. And sometimes like in Chrome or like in Safari, nothing will happen, but there like will appear some kind of notification that the pop-ups are blocked. So do allow them for this website, and the new window will open. Okay, who has done the enricher? Okay. And now, so are there any problems? Yeah, well, I believe people are getting the results here, more or less. So the enricher is a website, well, for enrichment, for pathway enrichment analysis, and it has a like very big and diverse collection of pathways, and like actually gene sets. And in particular, I really like it because of the transcription collection, which is so where you can try to understand which transcription factors are regulating your genes. So in our case, we can go, we can see that, for example, and actually here it's usually important to go to click on this and to see the table and p-values, and you'll see that there is a big overlap between our gene sets, like between our upregulated genes and this gene set that corresponds to IRF8 IRF8 transcription factor in mouse, in particular in BMDMs, and BMDMs are the like bone marrow derived macrophages is the same types of the cells that we have in our experiments. So remember, so our samples were BMDM untreated 1 and so on. So this is really good. So and this IRF transcription factor, we probably could say that, well, likely to be involved in LPS response, and I hope that it indeed does. We won't check it, but yeah. And there are also like some other gene set collections, well, with some weird ones like like NIH funded grants. So is there like any professor in the United States that study your genes? And so is there any competitions or whatever? Or like CRISPR, whatever. Okay, so this is, yeah. And also, like this, for this pathway analysis, there are a number of other tools that you can try to use. So, well, I used to like historically using M6DB tool a lot, and well, enricher for when I would like to look for some transcription regulation. But there are like web gestalt site is very straightforward and simple. And actually, here you can put your background genes, so like your gene universe, so the hypergeometric depth is done correctly. And there are other software, well, but I don't really like any other things. But WebGestalt you can try to use. Okay. And to conclude this part, so differential expression analysis, and well, it's like very simple and the most straightforward way to analyze the data if you have like two conditions that you want to compare. And pathway analysis allows you to go from individual genes, which is usually hard to argue. Like, well, you would like to argue about genes that are important, so that the genes that you can knock out. But usually there are many differentially regulated genes, and it's much easier to first argue about the biological processes, which can be done on biological pathways, which can be done through pathway enrichment analysis. And well, there are many ways how to do it, but the general idea is still that you are looking for the genes and for the you're looking for the genes in a pathway and try to understand if they are concordantly regulated, like in upward and downward manner. So if the pathway is upregulated or downregulated. Okay,